Hello everyone. So this video is about explaining lipid digestion and absorption. So how our body is digesting dietary lipids and how these dietary lipids are absorbed. So major composition of our dietary lipids is triacylglycerol. 90% of dietary lipid is 90% of that is triacylglycerol. So rest of the lipid content in our diet is cholesterol ester, it is phospholipid, it can be a free fatty acid, and of course there can be vitamin A, D, E, K. All these things need to be absorbed. So how the absorption process does? So the very first digestion of lipids will be going on in the stomach. So in the stomach there will be acidity. So in the acidity, so there will be an enzymes which are activated called lingual lipase and gastric lipase. So there are two enzymes. So let me write down the names of those, and that is lingual lipase and gastric lipase. So these two enzymes are acid stable enzymes. So you know the stomach has got hydrochloric acid. It's going to activate these enzymes and stabilize these enzymes. And these two enzymes, they are going to act on dietary triacylglycerol. So this triacylglycerol in the diet, it will be undergoing a breakdown process mediated by lingual and gastric lipase, and you're going to get fatty acids. Now one of the things that you need to remember with the lingual and gastric lipase is they are very specific for short chain fatty acids and medium chain fatty acids. That specificity you should remember. So SCFA, short chain fatty acids, and MCFA, medium chain fatty acids, these are the ones which are attached to triacylglycerol. They are the ones which are released by lingual and gastric lipase. You know the short chain and medium chain fatty acids are water soluble, so they can be directly absorbed by the enterocytes later. Okay, so this is what happens in the stomach. So the very first digestion of dietary lipids will be going on in the stomach, mediated by gastric and lingual lipase, which is helped by acidity in the stomach. Now when the gastric contraction occurs, so the things will get into the duodenum. So the first part of duodenum has got the endocrine cells, neuroendocrine cells present in the first part of duodenum. So they are going to synthesize and secrete two hormones, and one is cholecystokinin. So the very first hormone that we are looking at here is CCK, cholecystokinin, and the other hormone will be secretin. So cholecystokinin, what it does? So cholecystokinin will go to the pancreas and will pancreatic enzymes. Pancreatic digestive enzymes will come into the duodenum. Whereas secretin, what it does, it's going to bring bicarbonate. So these are the two main hormones that are involved in digestion process. So cholecystokinin releases pancreatic enzymes, secretin brings bicarbonate into the duodenum. Now the bicarbonate, it is going to neutralize acidic contents that are coming from the stomach, thereby the pH is brought up to 6 to 7. During this, at this particular pH, your pancreatic digestive enzymes, they are working at an optimal range. Why? Because they need alkaline pH. Now, which is the pancreatic enzyme that is responsible for digestion of lipid? And that is pancreatic lipase. So you need to remember here that pancreatic lipase, it needs a coenzyme. Basically it needs a partner called colipase, pancreatic colipase. Now this pancreatic colipase, it will come in as pancreatic procolipase. Pancreatic lipase, which needs a colipase enzyme, so that colipase comes in as pancreatic procolipase. Now the pancreatic procolipase is converted to colipase. That's an activation process, and that needs enzyme, another enzyme called trypsin. So trypsin is a proteolytic enzyme that's basically coming from pancreas as trypsinogen. Trypsinogen will be activated into trypsin by an enzyme which is located on the brush border called enterokinase or enteropeptidase. Once trypsinogen is activated into trypsin, that trypsin is going to activate pancreatic procolipase into colipase. and now this colipase, it's going to work along with the pancreatic lipase to digest triacylglycerols. Now, before all these enzymes work, so your lipid has to be emulsified. So there is a concept called emulsification that we need to understand now. So emulsification is basically, it is the mixing up of lipid with bile acids and bile salts. Now the bile acids and bile salts which are stored in the gallbladder, they will be brought to the duodenum by this hormone, that is cholecystokinin. So cholecystokinin causes contraction of the gallbladder and brings bile acids and bile salts. Once you get bile acids and bile salts, so these bile acids and bile salts, because they are amphipathic molecule, so they are going to interact with the large lipid molecule present in the duodenum. So this coating of bile acids and bile salts with the lipids, and then because intestines are contracting, so there is a segmentation contraction going on in the intestine, so that will give rise to breakage of these large lipid molecules into simple lipid droplets still coated with the bile acids and bile salts, and that entire process we call it as emulsification. So emulsification is nothing but it is coating of lipids with the bile acids and bile salts, contraction of the intestine, breaking it down into simple droplets called micelles. So we refer those lipid containing molecules which are coated by bile acids and bile salts as micelles. Now what is there in the micelles? So within the micelles we have triacylglycerol, we have tags within the micelles, triacylglycerols are present, there will be cholesterol ester in the micelle, there will be phospholipid present here, and there can be vitamin A, D, E, K. All these things can be present in micelles. These are undigested particles like triglycerides, cholesterol ester, phospholipid. They need to be digested. So how these things will be digested? So very first digestive enzyme I'm going to talk about is pancreatic lipase. So this pancreatic lipase, with the help of colipase, it's going to act on triacylglycerol present in micelles. Now there is a hindrance here. See, the bile acid and bile salt which is present over the surface of the micelle, it will have an inhibitory effect on pancreatic lipase. So bile acids will have inhibitory effect on pancreatic lipase, and that inhibitory effect need to be taken off. And who is going to do that? It is done by colipase enzyme. So what this colipase does? Colipase on one side it's going to interact with bile acids and bile salts on the surface. On the other side is going to interact with the pancreatic lipase enzyme. Thereby is going to take away the inhibitory effect of bile acids and bile salts on pancreatic lipase, and pancreatic lipase is now free to work on. So that's the use of having colipase. Now what is the function of pancreatic lipase? So pancreatic lipase is going to act on triacylglycerol that is present in the micelle, this triacylglycerol, and release free fatty acids. So triacylglycerols will be releasing free fatty acids with the help of pancreatic lipase along with the colipase. So what kind of fatty acids are released from the triacylglycerol? So it can release short chain fatty acid, medium chain fatty acids, and long chain fatty acids. All three of them can be released. But only one thing that you need to remember with pancreatic lipase is within the triacylglycerol. So if I write triacylglycerol here, 3 carbons and 3 fatty acids attached, say fatty acid 1, fatty acid 2, and fatty acid 3. So out of these 3 fatty acids, only fatty acid present in the first carbon, attached to the first carbon, and the third carbon. These 2 fatty acids will be released by pancreatic lipase. It's not going to work on the fatty acid attached to the second carbon. So it means at the end of action of pancreatic lipase, you are going to get 2 fatty acids and 1 molecule of 2 monoacylglycerol. And this is what is the digestive product of triacylglycerol in the presence of pancreatic lipase with colipase, and that will be there in the micelle. Now who is going to take care of cholesterol ester? So cholesterol ester is digested by another pancreatic enzyme called cholesterol esterase. So what this cholesterol esterase does? It's going to convert cholesterol ester into cholesterol plus fatty acid, and that will be going on in the micelle. Now phospholipid. Now the phospholipid is taken care by phospholipase A2. So this phospholipase A2 is a pancreatic phospholipase A2. It is going to convert phospholipid, that is PL here, phospholipid, into lysophospholipid plus fatty acid. So this is how it is going to act on phospholipid. So this will be going on in the micelles. So overall, at the end of action of all these enzymes, that is pancreatic lipase with colipase, cholesterol esterase, phospholipase A2, so your triglyceride is converted into 2 fatty acid and 1 molecule of 2 monocyl glycerol. Cholesterol ester is converted into cholesterol plus fatty acid. Phospholipid is converted into lysophospholipid plus fatty acid. So all this particular molecule, now it will be referred as mixed micelle. This is how you are going to convert micelle into mixed micelle. Mixed micelle. So mixed micelle, it has got 2 monocyl glycerol, it has got fatty acid, it has got cholesterol basically, not cholesterol ester, and it has got lysophospholipid, and it has got vitamin A, D, E, K. These are not changed. So these are the contents present in mixed micelle. So micelle is converted into mixed micelle at the end of action of pancreatic lipase with colipase, cholesterol esterase, and phospholipase A2. Okay, so this mixed micelle, so it has to be, it will, as it move down into the duodenum, so it will be absorbed into the enterocytes of the duodenum. So